Hello, my name is Jim Squires. It is a pleasure to welcome you all here today. I was invited and asked to talk about progressive familial intrahepatic cholestasis, and so that's what we're going to talk about. I think probably most of us have been in these types of forums in the past. I think for this one, what I would encourage folks to do is I believe you should have a chat option available to you or a place for questions. As I go through these, please put those questions in as soon as they come to you, and they'll be forwarded to me, and then we can address those either at the time if I see them or clearly at the end of the talk. And then obviously if there's any additional questions that come up as we continue to talk or at the end of the talk, we'd be happy to address those as well. So again, I think today we're going to talk about progressive familial intrahepatic cholestasis. So I always like to begin with a case. Again, I'm a clinician, I'm a hepatologist, a pediatric hepatologist here at the Children's Hospital of Pittsburgh, and so I think cases really tend to bring these things home for me. So this was a child that presented to us. It was a 3-month-old with a chief complaint of new onset jaundice or yellowing of the skin. From a past medical history standpoint, the family noted that maybe about 1 month of life, the child was squirming a bit at night, rubbing its eyes, but otherwise was developing and growing okay. Was taken to the PCP's office and diagnosed with reflux. Initiate a proton pump inhibitor or some acid suppression medication in order to treat that reflux, but really family noted no improvement in those symptoms of kind of irritability and squirming at night. Took back to the PCP and ultimately given a diagnosis of colic and sent on home. Parents then noted that the child began to have some complaints of loose stool and diarrhea, and so brought the child to the ED. And it was really here that the physicians caring for the child really first noticed that the child was jaundiced on exam. They did go on to get some screening labs of the child. And again, for those who aren't clinicians, this is kind of the shorthand way in which we present some of these labs, but this is the complete blood count showing a relatively normal white count at 5.2, a hemoglobin at 10.1, and platelets that were elevated at 492. From an electrolyte standpoint, everything looked pretty stable. The child did present a little bit dry with a BUN and a creatinine of 32 and 0.8, which are a little bit high, again suggesting dehydration and consistent with the history of diarrhea. From a liver studies standpoint, these are the liver tests that were sent. So this is the ALT and the AST, 2 tests of hepatocellular injury, and those were elevated at 234 and 167. GGT, which is kind of more classically a marker of cholangiocyte injury, was normal, but can also be a marker of cholestasis, and we'll come back and talk about this in a little bit. And then the bilirubins, the total and direct bilirubin are the numbers at the bottom, 6.8 and 5.7, clearly elevated in this child. From a function standpoint, as far as how the liver was actually functioning, it looked okay. Here are the coagulation studies with an INR of 1.1. But it's really these labs here that were probably most important to focus on. And so this is the total bilirubin and the GGT. And this kind of clinical picture of an elevated total and direct bilirubin level with a relatively normal GGT is highly suggestive of what we're going to talk about today, but we'll get to that here in just a second. And so when you think about, again, kind of cholestasis in a child who presents to the office, right? I think historically, the differential, right? The list of things that can cause cholestasis in a baby, it was pretty small, right? You go back to the 70s, there were about 4 things you really had to think about. Biliary atresia, which is a rare cholestatic liver disease that's important to recognize early because there's an intervention. We're not going to really talk about that today. Infection, right? So the torch infections is a family of infection that we typically think about in neonates. Miscellaneous, which is kind of a collection of known diagnoses, but were very small, that only make up about 7%. And then the vast majority of kids, again, back in the 70s, had what we called neonatal cholestasis. And so neonatal cholestasis is just a descriptive term of a neonate with cholestasis. But the reason behind that cholestasis really wasn't identified. Over time, we have really been able to shrink that pie piece down of neonatal cholestasis into known diseases, known diagnoses, many of which have a genetic basis, and we'll talk about some of that today. But again, you can see again as of 2020, the piece of the pie that is still biliary atresia hasn't changed. That's at 25%. But you see that neonatal cholestasis pie has really decreased as we've been able to identify diseases such as alpha-1 antitrypsin deficiency, various metabolic diseases of different types, and then the intrahepatic cholestatic diseases, which is mainly where we're going to focus our talk today. And so, what are intrahepatic cholestasis diseases? So, more broadly, if you think about what are diseases in children that cause cholestasis that have a genetic basis, this was a review that Pat McKiernan and I put together now about 5 years ago that really looked at all of the molecular mechanisms that can cause cholestasis. Focusing specifically on those PFIC cases, you can see here of what 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 genes that we were able to kind of discuss and identify and report in this paper that had been associated with progressive familial intrahepatic cholestasis, or PFIC, and we'll come back to that. So if you go back to kind of our kid, right, our kid that presented to us, this is kind of historically how we would approach that child. Right? Someone comes in with neonatal cholestasis. One of the first things you want to do is make sure they do not have biliary atresia. There's a surgery or an intervention that we then pursue in those kids, and it's timely, and so you really want to kind of rule that out early. Obviously, alpha-1 antitrypsin deficiency is a disease that's been around for some time, and so there is a relatively simple phenotype that you can send to look for that disease. But then, you're getting into that, obviously, comprehensive family and patient history to look for red flags. You're looking for treatable conditions. And most of the time what ended up happening is you kind of go on this diagnostic odyssey where you'd send any number of lab tests, imaging tests, biopsy tests, all different kinds of tests to kind of rule in or rule out different diseases. And one of the last things that you did was genotyping or genetic studies. Now, this has really, kind of evolved over time. This has paralleled the accessibility of genetic testing and the cost as these tests have become much more cost efficient to send. And so now newer algorithms, have really reshuffled the algorithm. And the reshuffling has mainly been in the form of how we approach genetic testing in these children, and that it has moved much further up the algorithm, which is if it's not BA, if it's not alpha-1, we send genetics, right? That's kind of, in a very simplistic form, how these children are approached today in the clinic. So if we go back to our kid, we did get some imaging. The liver was noted to be heterogeneous, a normal gallbladder, a normal bile duct. That's very reassuring for it not being biliary atresia, where you typically see abnormalities. We did send the alpha-1 antitrypsin phenotype, which came back PIMM, which is normal. So this child did not have alpha-1 antitrypsin liver disease. And so the next thing we did was we sent off one of our fancy targeted gene panels. These are, these targeted gene panels are ways in which we can test for a collection of different genes, often in a family of proteins that share a same phenotype. So in this case, this is those that are associated with neonatal and adult cholestasis. There are targeted gene panels for, seizure disorders, for, any number of, ciliopathies, any number of disorders where there's a similar phenotype. You can send these targeted gene panels. As it relates to PFIC, in this particular panel, although I think there were 10 in that paper that Pat and I put together, this panel was testing for at least 5 of those genes. We sent it off, and then this is what came back. This was the report of that child, showing here again that there were 3 different genes that did have defects in them, but most importantly, this ATP8B1, which does associate with progressive familial intrahepatic cholestasis type 1. You go up there to the top, you see ATP8B1, apparently homozygous. That's pathogenic. This kid has PFIC1. So we now have a diagnosis. We go to the family. We say your child has PFIC1. This is what the family asks, right? My gosh, what is PFIC1? How is it treated? And what does this mean for my child? And so these are some of the things that we're going to try to touch upon today with these objectives, right? We're going to define the current understanding of PFIC. We're going to delineate some of the gaps in current knowledge, right, and where things need to be filled in. And then we're going to kind of define a little bit opportunities for future collaborative research. And then along the way, we'll also discuss treatment options and other ways that we manage these children in the clinic. And again, if anybody has joined late, I want to welcome you, and then just only say that if there are questions that pop up, I think please put them in the chat as soon as they come to you. They'll be forwarded to me at some point during the talk, and we'll make sure we answer them at the end, and we'll go from there. So, how do we define our current understanding of PFIC? So I really want to kind of focus on these kind of, 3 of many review articles, but 3 that I'm familiar with. The first 2 I kind of helped co-author. The first one led by Rodrigo Florentine, who is a PhD here at the University of Pittsburgh working in PFIC research. The second one, Sarah Hankel, one of our former transplant fellows, had put together. And then this last one, most recently published in Hepatology, led by Summer Ibrahim, who's at Mayo, and Saul Karpin, who's down at Emory, kind of looking more broadly at some of the cholestatic liver diseases with a genetic etiology. Specifically to PFIC, there's kind of, 2 different buckets that people kind of will put PFIC diseases into. Again, PFIC is a descriptive phenotype. PFIC stands for Progressive Familial Intrahepatic Cholestasis. That just describes a phenotype. And historically, there were 3 of these disorders that we talked about. There was PFIC 1, 2, and 3, and that was it. There's now what we call the expanded PFIC, and I'll just say that's PFIC greater than 3, because although I think there were 10 that we reported in that paper, there were 5 on the gene panel. I think now there's probably 12 different genes that are associated with that. We're going to talk about some of this in a little bit, but we kind of think of historical and expanded PFIC. So what about these historical PFICs, these PFICs 1 through 3? So there are some notable differences in these, and again, some of these papers are a little bit older because these are the PFICs that have been around for the longest. These are also the PFICs that are probably the most common. And there are some differences, some notable differences between these 3 historical conditions. So PFIC 1, you can see here the age of presentation is typically in infancy. End-stage disease is relatively rapid, developing over the first decade of life, again, making that course moderately severe. Pruritus or itching can be significant in these kids. It can actually be devastating. It can be, they can really mutilate themselves. You do have extrahepatic manifestations in PFIC 1. In this child that we presented at the beginning, the diarrhea was a clue for PFIC 1, as these kids do have intestinal disease associated with their deficiency. They do not have any risk of liver tumors as far as we know. Again, cholesterol stones, I won't go through the labs, but there are some kind of notable differences. PFIC 2, again, presentation can be in the neonatal period, but oftentimes a little bit later, early infancy. Again, the disease course can be rapid within the first few years of life, and making the course quite severe. Again, pruritus can be refractory and really one of the main complications of this disorder. They do not have extrahepatic disease because the BSEP protein is solely isolated to the liver itself, and they do actually have quite a high risk of liver tumors. And so monitoring and screening for liver tumors in this population is something that we always need to consider. And then PFIC 3, these patients typically present much later in life, sometimes even into adulthood. The progression is typically much slower, first or second decade of life, if it is progressing. The disease course can be a little bit more insidious. Pruritus is there, but maybe not quite as refractory or severe as some of the other PFICs, or definitely some of the other historical PFICs. And again, so these top 3 boxes really kind of highlight, some of the clinical differences that we have thought about or think about when we are seeing these patients in the clinic, particularly when they present with us with new mutations or as a new patient. So I do want to kind of focus again a little bit on these historical PFICs because of there is a body of knowledge that we understand about them, and we'll first kind of talk about PFIC 2. I start with PFIC 2 because it's kind of the simplest of the PFICs to understand, right? Again, the idiopathophysiology, the ABCB11, that's the gene that associates with this. The protein is the BSEP protein, and the disease is PFIC 2. BSEP stands for bile salt export pump, and so this is pretty simple. This is a pump, and the way this pump works is that it takes bile salts that are made inside the liver cell and it pumps them outside the liver cell. So it's a pretty simple to understand the mechanism of the PFIC protein. Again, it's the best understood. It transports these bile acids from inside the hepatocyte to outside the hepatocyte. And there are various, as with all of these, genetic mutations that can either lead to totally absent or a dysfunctional protein. And so there is a variability in phenotype that we will come back and talk to about a little bit further on in the talk. What is the histology of PFIC2? It's relatively ubiquitous, right? You kind of get this giant cell hepatitis that's associated with it. But I think most importantly is there is a BSEP stain that can be done. And you see in B and C, in B, that stain is totally absent. In C, you kind of get this disarray of brown. When under normal conditions, this is a control, you can see that you get this very kind of fine linear staining of the BSEP protein, which is typically in the canalicular space. And so that is important. And at least historically, before there was genetics, biopsies were very helpful for BSEP deficiency. And that although the initial kind of H&E might not have been diagnostic, the BSEP stain was at least fairly suggestive of this disorder. So we will move on to PFIC3 because, again, we know a little bit more about this protein than some of the others. The protein is the ABCB4. The gene is the ABCB4. The protein is the MDR3, and the disease is PFIC type 3. So again, this protein is somewhat understood. It acts as what we call a phospholipid translocator flippase. The thought here being that, again, these bile salts that are made in the liver and trying to get out, the function of these bile salts is to act as detergents so that we can absorb our fats. I think we've all done the experiment in grade school where you pour oil and water and they don't mix. So when we eat a big fatty meal, getting those fats across a membrane or across a structure that is mostly water can be complicated and requires detergents to do that. So that's what bile salts do. But if you also think about, on a cellular level, every cell is surrounded by a lipid membrane, right? A very small lipid membrane that is made of fats. And so if we're making these substances inside a liver cell, whose job it is to help us, break down and absorb fats, you have to be very careful about how we traffic those things, because if they intermix with the cell itself or the cell membrane, you can get destruction. And so what the MDR3 protein does is it essentially secretes phosphatidylcholine. It flips it from inside the cell to outside the cell as these bile salts are moving through that cellular membrane so as to buffer the action of the bile salts against the membrane itself. So you can think of this again as a way to kind of protect the cell as bile salts, which can be toxic to the cell, are being trafficked across the membrane. So it neutralizes the detergent activity of these bile salts at the cellular level. And then there's PFIC1, right? PFIC1, again, what the child in the beginning was diagnosed with, ATP8B1 is the gene, FIC1 is the protein, and PFIC1 is the disease. So this is the least understood protein, right? We know that it's a multi-span transmembrane protein. We think there's actually a couple mechanisms that have been tagged to it, but one of the mechanisms we think it acts is as what we call a floppase. And this is essentially similar to what the MDR3 protein does. We think FIC1 also acts to kind of flip certain constituents of the lipid membrane in order kind of to help protect against those bile salts as they're coming across the membrane. But, it's not entirely clear that this is the main function or even the only function of this protein. So at the end of the day, we still don't really know what the FIC1 protein does. And we'll come back and talk about how we're trying to understand that a little bit more towards the end of the talk. And so, those are the three historical PFICs, right? So what about these expanded PFICs, right? I always like this because, this has really kind of exploded over the course of my career. I have a past kind of like, 5 to 10 years. So I had been in 2017, when I had, been here for a couple of years in Pittsburgh, I was asked by one of our adult research groups called the Pittsburgh Liver Research Center to give a talk on PFIC diseases. And this was one of the slides from that talk, right? So again, you see the PFIC slide. Historically you had these three, and now we have these two more, low GGT cholestatic conditions, right? So as of 2017, we had only known of 2 additional ones. I just mentioned that there was at least now 9 additional ones. And so this has really been an explosion of our understanding of the genetic underpinnings of neonatal cholestasis. So that kind of leads us into the next part of the talk, which is where we will delineate some of the gaps in knowledge of these disorders. So again, I think looking at how we used to define cholestasis back in the 70s to how we've defined it in this decade, clearly a lot of progress has been made. But I think, if you focus on this, kind of 25% of intrahepatic cholestasis, and, there is still this 15% of neonatal hepatitis, right? This is a wastebasket term for what we don't know. I don't know what's causing the cholestasis, but clearly something is driving this child to have cholestasis. Probably today we've chipped away at this number, right? Even from 2020 to where now this is now closer to the 10 to 12%. And this is why, right? Again, back in 2014, progressive familial intrahepatic cholestasis, which is where that table that I had presented at the beginning came from. Really, there was just the 3 PFICs. There weren't any more than that. Kind of further on, that was in March of 2014. In April of 2014, right after that review came out, this paper in Nature Genetics came out describing TJP2 as a genetic basis for progressive cholestatic liver disease in neonates. And so this was then kind of named PFIC4. TJP2 is what we call a tight junction protein. It's also called zona occludens 2. These tight junctions are really kind of essential for the liver because, again, if you think about those bile salts that are getting out and then trying to get out of the liver, you don't want them to kind of leak back between cells. And so you need tight junctions to keep cells together in order to prevent things from kind of leaking back through. If you have defects in a tight junction protein, you can get a reflux of bile salts back into the intracellular space, which can cause a phenotype similar to some of the other PFICs. And so TJP2 came out in 2014. A couple years later, in 2016, this paper came out looking at mutations in the nuclear bile acid receptor FXR, which was then reported to cause the phenotype of progressive familial intrahepatic cholestasis in another small cohort. Again, FXR is coded by the NR1H4 gene, and this was kind of deemed PFIC type 5. FXR, as some of us may know, it's a nuclear receptor. It's activated by bile acids and directly involved in the expression of both BSEP and MDR3, some of those proteins that cause the PFIC 2 and 3. And so if you have an upstream regulator of BSEP and MDR3 affected, you might not be able to traffic those proteins appropriately to their location. You might not be able to make those proteins correctly. And so again, you can get a similar phenotype of PFIC, even though those particular genes, they don't have a defect in them. It's a kind of a modifier gene. 2017, a year later, Myo5B was reported to cause neonatal cholestasis, or at least a low GGT cholestasis in a child who didn't have microvillus inclusion disease, which is the disease that Myo5B has historically been associated with. And so, this was, again, PFIC 1, 2, 3, 4, 5, and 6. And so this was then when Dr. Henkel, Sarah, and I put together this review where we tried to kind of sum up what was known at the time of that publication back in 2019. And so this is, what was our understanding at that time of how these PFICs work. So you have your historical PFICs there along the bottom, FIC1, BSEP, and MDR3, BSEP being the bile salt pump, MDR3 being that flipase that is flipping phosphatidyl cholines outside the liver. FIC1 may be acting as a flipase to reconstitute the membrane at the intracellular level. And then you have the 4, 5, 6. You see on the right 4, the TJP2, again, acting on the tight junctions of the liver cell. When there's defects with that, you get bile salt leakage back through. You have the FXR protein up there in the nucleus, which is acting on the promoter region of the BSEP gene. So if you don't promote BSEP being made, you're not going to get BSEP. So that's why the phenotype is similar. And then you have the Myo5B, which is that big thing there in the middle. Myo5B functions as a protein that recycles the BSEP protein within the hepatocellular membrane. So obviously, if that process isn't working very well, again, the end result is a somewhat dysfunctional BSEP system, even though the BSEP protein itself doesn't have the defect in it. So again, this was 2019. About 3 months after we published that paper, this paper came out, which reported 5 additional genes associated with the phenotype of progressive familial intrahepatic cholestasis in children. And then, as of 2020-21, ABCC12, Alex Mithke in Cincinnati, put this paper out in Gastroenterology, which is probably the most recent PFIC that's been described. It's one that makes your head explode, right? I mean, the evolution of this field over this short time frame has been daunting to all of us who are trying to care for these kids and these various disorders. And so, this kind of editorial that was put out by Paul Manga from the Pittsburgh Liver Research Center, looked to kind of question that, as many of us had done. Is it time to transition to to genetic cholestasis, right? Leave this term PFIC in the dust, and really kind of more define these based on their genetic basis. Because one of the things that they pointed out, right, is identifying a single gene is really insufficient to account for the phenotypic variability for establishing mechanistic principles in many diseases. And so, one of the issues when thinking about mechanistic principles, right, there are a number of PFIC proteins that we know have been identified to cause the PFIC phenotype that we don't actually know what these proteins do, right? I mentioned FIC1 already, right? I mean, we kind of know a little bit about FIC1, but we really don't know what its full function is. Some of these other ones, USP53, LSR, WDR83OS, when you look at the literature, we don't know what these proteins do. And so they're being reported to cause the PFIC phenotype, which is great, but I can't even tell you what this protein does on a functional level. And so it's very challenging to know how to care for that, or even think about how to care for a child with these conditions, or frustrating to have a conversation with a family when we say, yep, we got the protein, don't know what it does, don't know what we can do to help you. And then there's this aspect of phenotypic variability, because even within the same family of disorders, right? Just the PFIC1s or just the PFIC2s, you can have incredible variability of the phenotype of these kids. And so we'll talk about that here in just a second. So as an example, USP53, and I start here because it's kind of the smallest numbers to understand. So worldwide, only 31 patients have been described. This is incredibly small numbers of patients who have been described with these disorders. All of them have a low GGT cholestasis and pruritus. Again, that kind of classic PFIC phenotype that we talked about. But the presentation can be from infancy all the way through adolescence, right? So it's not as easy to identify these kids as maybe it was the 1, 2, and 3. Some of them have extrahepatic manifestations like hearing loss, others do not. Cholestasis in some of these kids has been reported to be severe, refractory, progressive. Some of these kids have gone on to need liver transplant, and others, it's very mild. Right, and you start them on, kind of very, generic supporting type medications, and the numbers seem to get better and the symptoms go away. Even though on paper and at the genetic level, it looks like they have the same disease. I think BSEP is where we are really kind of learning a lot more about the genetic variability, even within the same protein. And a lot of this has come out of what we call the NAPID group. The NAPID group is a European coalition that has kind of retrospectively looked at this. They have partnered with many of us here in the United States, particularly through a network called the Children Network that focuses on these diseases. Hankin Verkad, who's the senior author on this paper, has really kind of led this effort. So in this initial paper, which came out in 2020, they wanted to look at the genotype and how that correlated with the natural history in BSEP deficiency or PFIC type 2. And what they found was that which defect you had in the protein correlated with physical manifestations or phenotypes of these disorders. And so he began to kind of subdivide BSEP into BSEP 1, BSEP 2, and BSEP 3. BSEP 3 was the most severe. That's the kind of dark blue line. And these are patients who, when you looked at what type of mutation they had a truncating or a non-functional protein result from their mutation. And so you see in the top bar, the percentage of those patients who were alive with their native liver was 20%. Right, at 15 years of life, so very, very, very low. And if you look at the percentage of patients with carcinoma, these were also the ones that were most likely to develop cancers, right? Upwards of 30% at 15 years after diagnosis. If you looked at whether those patients either had none of those mutations or one of those mutations, you saw that they seemed to do, not the truncating, but some of the more common mutations, this P D4A2G or PE297G, these mutations, which seem to be more common when we do genetic testing on these kids, seem to have a little bit of a better phenotype, right? Suggesting maybe that these were dysfunctional proteins, so they were there but just not working very well, versus not there or not working at all. And so that, again, even within the same group of patients, there is phenotypic variability. This same group has gone back now and looked more specifically at some, okay, I should say, I've kind of built on those initial findings. And here what they did is they again looked at those BSEP 1, 2, and 3s. The ones are the BSEP patients that tend to do pretty well with these two particular mutations that are common in the community. And what they looked at is they said, well, what if you have none of these? Again, that's BSEP 3. We already know that those people did bad. But what if you have one good and one bad mutation, right? So that's that middle box there, the BSEP 1, BSEP 3. What they found there, interestingly, was that those patients did just as bad as the BSEP 3s, really suggesting that these more severe phenotypes are the drivers, right? So if you're heterozygote but have one bad mutation, you're really going to have a bad outcome. And so all that is to say is that we will call these children all BSEP deficiency, but they have very, very different clinical paths based on their exact genetic mutation. And so as we are learning more about the genetic contributions, we are understanding that we need to even subdivide rare diseases into different potential populations. Again, PFIC1, there's a lot of variability in PFIC1, and we've clearly appreciated that here. This was a paper that we put out now several years ago that looks at clinical variability after what we call external biliary diversion. So one of the historical treatments for these disorders, or at least one of the reasons why we think these kids get such severe liver disease is, again, it kind of comes down to bile acids, right? And so if we can somehow manage the bile acids, maybe we can improve their phenotype. And so this is how bile acids typically are made and evolve in our system. They're made in the liver, they're excreted into the liver, into the small intestine. Most of them, again, are used to help us break down and absorb fats, and then those fats and bile salts get back to the liver. Those that aren't used to reabsorb fats come down to the terminal ileum, where there is a protein, the IBAT, the ileal bile acid transporter, that then reuptakes that bile salt, goes back into the liver, and this is called the intrahepatic circulation of bile acids. And so the thought is that for those bile acids that are being reabsorbed in the terminal ileum or the intestine, if there is a way that we can prevent them from coming back to the liver, building up and causing damage, potentially we can improve the phenotype of these children. And so historically, how we have done that has been with a pretty blunt tool of biliary diversion. Biliary diversion is a way in which you divert bile, again, away from that normal intrahepatic circulation and don't allow it to come into contact with that ileal bile acid receptor that's only located in the terminal ileum. There are three classic ways to do a diversion. One is called the partial external biliary diversion, which is the A picture there. You can see they take a loop of bile, they tack it up to the gallbladder, and then they bring it out to an external stoma, so that bile then leaks out of your body instead of going into the intestine. Then you have the partial internal biliary diversion, where you take the cap of the gallbladder and tack it to the transverse colon coming across, again, so it doesn't come in contact with that terminal ileum section. And then you have the ileal bypass, which is represented by picture C. I won't specifically talk about all the pros and cons of these different approaches. I'm happy to take questions if there are any, but when we talk about biliary diversions, these are the three kind of classic ways that it has been done. Most centers have their just institutional experience. There's never been any comparative effectiveness trial that has looked at different biliary diversions to determine which one may be better than the other. So that data is not even available. But our experience here in Pittsburgh is that we have classically done the external biliary diversion. And when we look at our kids with PFIC1 who have had external biliary diversions, what we see in their post-diversion course is a lot of variability. Some patients do very well. They clear their bilirubin and it stays down, doesn't come back. Some patients will clear their bilirubin initially, but then have these intercurrent episodes of cholestasis. Oftentimes that can be associated with like other illnesses and things that these kids get. Some kids have cleared their bilirubin initially, but then have developed cholestasis that's come back and then hasn't resolved. So, even though these kids have the exact same, and these kids in particular, like our population is very genetically homogeneous. We take care of a very small slice of PFICs, in that it's typically the Amish community that we draw our patients from who have this disorder. They all have the exact same genetic mutation, and yet their phenotypes can be incredibly variable. This has also been looked at on a larger scale. Again, this comes from the Children Network, the Children Liver Disease Research Network, which is kind of an NIH-funded consortium that looks at many cholestatic liver diseases, including PFIC. And when Casper Wong, who was in Southern California, looked at the effects of surgical interruption, right? The various ways in that we surgically intervene on these kids and how that affects these disorders. What he saw when looking at the PFIC1 and the BSEP, right, is that, again, you have pre-op and you have 6 to 12, 12 to 24, and 24 months after their surgical intervention. And this is the distribution of pruritus, whether it was absent, mild, moderate, or severe. So going in, almost all the kids either had severe or mild to moderate pruritus, right? Again, it's one of the dominating features of the condition. They get diverted, and you can see after diversion, there is variability in how they respond to that surgical interruption. So it wasn't perfect, and there's still variability. And again, this most recent paper looked at the impact of genotype on and serum bile acids on diversion in just PFIC1 deficiency. Again, this is that NAPID group. They had various PFIC1 populations, similar to what they did in the BSEP. They tried to look at the genetics of the PFIC1 and separate them into only missense mutations, predicted truncating mutations, or 2 predicted truncating mutations. And here, what was interesting is there actually wasn't any difference, right? So even if you were predicted to have a very severe mutation versus maybe a more benign or a more I should say less severe, right? None of this is benign. A less severe mutation. All of these kids still had, the same outcomes when you looked at percent of patients alive with the native liver or the percent of patients, after their biliary diversion. Again, just building this case, right? And so, what they found here was that, the number of predicted protein truncating mutations did not associate with natural history or prognosis of PFIC1 deficiency. So as opposed to the BSEPs, the PFIC1 maybe not so much, but as I showed you before, from a phenotypic standpoint, there is still a lot of variability. So it just gets complicated. And we're learning more and more each day about the complexity of these various disorders. So what about some of the gaps, right? So treatment is a huge issue, right? Historically, treatment has been mainly supportive. We really haven't had anything to offer these kids. We would do the diversion, but whether or not that really made an impact, I think was still a little bit of an open book, and not every center even offered the diversion, right? IBAT inhibitors, which we'll talk about here in just a second, versus biliary diversion versus transplant. Who do we do it in? When do we do it? Which type do we do? Why? We didn't know any of this, and oftentimes, we still don't. And so it can be very challenging and frustrating to care for these patients because they can be so severely sick. And oftentimes we aren't able, despite all of our knowledge and exploding knowledge of the genetic basis of these diseases, very little, to be honest, has changed about how we treat them. The one exception has been with these IBAT inhibitors. So again, if you go back to this picture, this is how bile acids have historically not historically, this is how they circulate through the body with the intrahepatic circulation. We talked about that IBAT protein there in the ileal cell, the terminal ileum. These newer medications, what they do is they essentially block the IBAT receptor or protein in the terminal ileum and prevent those bile acids from reabsorbed. And essentially, you eliminate them in the stool. So this is essentially a pharmaceutical way to do a diversion. And so this has changed how we treat these patients. We do have these approved now for these conditions. They have been impactful, mostly as it relates to decreasing itch and decreasing circulating bile acids that are measured in these children. These were only been approved over the last 12 months or so. And so what we don't really have is great longitudinal data to know if these are going to actually change the natural history, right? Are they going to actually extend native liver survival? What we don't know is if they're going to augment surgical diversion or other therapies. Again, we're all kind of learning with these new medications as they become approved. But it's exciting now that we actually have a medicine that is targeted to these diseases that I really hope will be impactful for these populations. And so again, I want to kind of use the last bit here, and hope to end with plenty of time for questions if there are any, to kind of define and discuss a little bit about opportunities for future collaborative research. And I want to focus a little bit on this paper here. Again, Alex Meeke, who's one of my former colleagues back in Cincinnati when I did my training there, because I think this really highlights any number of ways that we can think about our understanding of these kids and opportunities for discovery. So in this paper, right, they had a kid, again, another case, 1-year-old, presented with a normal GGT cholestasis. And again, that low GGT, normal GGT cholestasis, that's kind of a red flag for all of us, that there is likely a genetic basis for this kid's cholestasis. We just need to uncover it. So had a very extensive workup, including that gene chip testing that we talked about. And again, the gene chip that I showed initially has expanded now to where most of these PFIC genes that we've talked about are now included in the chips. That test came back negative. They then went on to do whole exome sequencing. Again, I think these targeted gene panels, test for genes that we know associate with a phenotype. They don't test for every gene, and so when the initial screening genetic test came back negative, Alex sent out the whole exome sequencing, and here it was identified as a homozygous truncating pathogenic variant in this ABCC12 gene. What is ABCC12? So it encodes for an ABC transporter, MRP9, with an unknown function. So here we go again, right? Well, we now have another gene that we think is going to associate with neonatal cholestasis, and we don't know what it does. But what I like about Alex is that he's a continually curious individual, and so he said, okay, we need to figure out more about this, right? So this is the liver histology of the child, and what you saw was that this histology revealed that the injury was really defined by extensive ductular damage, the bile ducts themselves. And I think you can appreciate that most probably in picture B here, right? In a normal liver biopsy, you kind of see a very nice circle similar to this red circle here of this brown stain. This is the CK7 stain. And what this shows is that there's complete disarray of the cholangiocytes or the bile ducts that make up the system that's trying to drain the liver. And so I think as opposed to, again, C, which is a control, that's a normal bile duct with an immunohistochemistry. In D, which was their patient, when they looked for that CK19 stain, that stain that defines the bile duct cell, they really saw none of it. And so, what you were seeing histologically was that the bile ducts seemed to be severely affected in this disorder. So then, they went a bit further and did targeted Sanger and next-gen sequencing of the ABCC12 gene in a cohort of 89 low GGC cholestatic kids. So again, this was likely in collaboration with the Children Network, where there's a collection of these kids who we know have a phenotype but don't necessarily have a genetic cause yet, and they were able to get 89 different samples. So here are the 89 kids. They're all cholestatic. They all have a low GGT cholestasis. And these kids really belong to two cohorts, right? So cohort 1 consisted of 34 children with a low GGT cholestasis, in whom the gene chip had been performed, that targeted gene panel. And then the cohort 2 was 55 children enrolled into, again, the children's study. None of these were known to harbor any other disease-causing mutations. When they did the targeted sequencing of their new gene on this population, they found that 6 variants were identified. 2 predicted to cause premature truncation, and 4 variants of unknown significance in, again, 6 additional kids. All right. So again, what is MRP9? I think, when they looked at the immunohistochemistry, it revealed that MRP9 protein, and this is in, again, controls, right? This protein is located to the large and small ducts, typically in the zone 1 hepatocytes in some of the control tissue. And so, this typically seemed to be more associated with cholangiocytes. When they did immunofluorescence, it showed that MRP9 was modestly enriched in the apical membrane of the interlobular bile duct. Again, this is a cross-section of a bile duct inside of a liver cell, and you see that really neon green staining is for the MRP9, and you see that it kind of localizes to the inside of that circle near the apical membrane. They then took this information and went to a zebrafish model and found that in a zebrafish, the ABCC12 was expressed again in the intrahepatic bile ducts, again, the stain is the neon green sign on the right, and it's overlaid with the stain is the red stain on the left, overlapped with the markers for the cholangiocytes on the left, and you see that they very much mirror each other. And then they also looked in mice, right, in a mouse model as well, and showed that in Western blot, the MRP9 was modestly located within the cholangiocyte. I think you can see that up top here. In the mouse, these are the cholangiocyte cells. You can see that there's a dark bar here that suggests, that suggests that this is also in the cholangiocyte of a mouse, right? So they're building a case for this protein to be located within cholangiocytes in 2 different models. And so what they kind of concluded was that MRP9 did show evolutionarily conserved expression in cholangiocytes in 3 different species, the zebrafish, the human, and in the mouse model. So then what they did is they went back and they said, hey, well, what if we use some of this new fun CRISPR-Cas9 technology and try to create this as a new animal model? And so they took zebrafish and they took mice, and they essentially used CRISPR-Cas9 technology to knock out MRP9 in those models. And what they collectively saw was that the evidence suggested that MRP9 deficiency in 2 animal models demonstrates that a lack of it confers susceptibility to a loss of intrahepatic bile ducts, similar to their patients. And again, I won't necessarily go through all the data, but you can see that those who have are knocked out have higher bilirubins and markers of cholestasis. You can see that the bile salts are higher in those organs because, again, bile salts aren't able to be excreted. And you look at histology, what you don't see, or at least trust me, I can tell you what you don't see, is a normal bile duct, right? And so, this is kind of the kind of summarizing picture of their model, right? In that they had a child, that kind of came in that didn't know what the kid had. They found that there was a protein there. They didn't know what the protein did. They then went into the lab and said, okay, is this protein located in animal models? It was. Is it located in the liver? It is. And then what happens if we knock this out in an animal model? Does it recapitulate the phenotype? It does, right? So that is kind of, I think how we need to think about opportunities for exploring these kids in pipelines like that into the future. So again, what are some of these opportunities, right? Incredible advances are still needed. As noted, these are incredibly rare diseases, right? Only 31 patients published in the literature with TJP2 deficiency, with the USP53 deficiency. But what this case demonstrates is that you don't need a large patient population to really make an impact, right? You really only need a few in order to kind of change the landscape. Just because there is a lack of established animal models doesn't mean that it's going to be a huge barrier, but we're going to touch on this in just a second. And you need to have close collaborative relationships between clinical and research teams. But again, this has been one of the historical barriers to all of this, right? I think, Alex's example was great because he was able to make a couple animal models that did recapitulate the disease. But for the most part, mice don't equal humans. And this is particularly true of many of these PFIC disorders, because although Alex's model, we still don't know what the protein does. We know where it that it localizes to the cholangiocyte, and they showed that when you knock it out, you get kind of bile duct paucity. We still don't know exactly what that protein does. For some of the other disorders like BSEP and others, we know what the protein does. The challenge is that in an animal model like a mouse, they have a different system of eliminating bile salts from their livers. And so there isn't a way necessarily to knock out, for example, a BSEP or a PFIC1 or an MDR3, because they have compensatory proteins that when we've tried to do that historically, will upregulate and not recapitulate that phenotype. And so we have to think about other ways outside of what Alex did even to further explore and study these populations. And so, again, what are some of the unique things that we have here at the University of Pittsburgh? Well, one of which is our location. We are conveniently located between, almost evenly, between Lancaster, Pennsylvania and Middleburg, Ohio, which are the 2 largest Amish Mennonite community populations in the United States, where diseases such as PFIC1 particularly are incredibly prominent. Many of you may know that PFIC1 is also called Byler's disease because Byler is the family name of an Amish Mennonite family group that this disease can be much more prevalent in. And so we have a number of kids whose last name is Byler that we care for who have PFIC1 deficiency. And just to kind of put that a little bit into perspective, right? This paper that came out from the NAPID group that was really big, they had 130 patients in, I don't know how many co-authors are there, right? But 40, 50 centers, right? Collaborated to come up with 130 patients. I counted ours. We have about 30 to 40 Byler's kids that we care for just at UPMC. So we have, although still rare, a relatively huge number of patients with this condition that I think gives us a unique opportunity to explore. And I've been working with this gentleman, Alex Soto Gutierrez, over at the Pittsburgh Liver Research Center to do additional studies to look at how we can explore this. And so we had put together some funding to do a study looking at the potential use of human-induced pluripotent stem cells in the study of these diseases. Currently we've enrolled 26 subjects, 16 subjects we've collected tissue on and have produced stem cell lines in, and these include patients with both PFIC 1, 2, 4, as well as VSPS 33B, which is ARC syndrome and another type of intrahepatic cholestasis disease. And in short, just to kind of give you a very high-level view of what we're trying to kind of do, right? What Alex has been able to do is essentially show that, what we do is we take a liver cell, we back it up into a pluripotent form and then reprogram it to become a hepatocyte-like cell. And when he does that, he can show that, again, on A, what you can see is that the mutation is conserved in the hepatocyte-like cell, and that what B, C, and D show is that these cells also recapitulate other forms of both hepatocyte cell lines and inducible pluripotent hepatocyte that have been made. And so these FIC1 cells that are recreated to be hepatocyte-like cells potentially can be a mechanism to recapitulate and study these disorders at a bench level. And so this is very exciting. And this is kind of, the pie in the sky way which we think we might be able to do that, right? Is have a patient through a variety of different mechanisms, be able to study that patient both at a cellular level and potentially in animal models, and then use these models for both concepts of cell therapy and concepts of disease modeling and drug discovery. And so these are the big picture ways in which I think hopefully we'll be able to change the landscape for the care that we provide to these patients in the future. So in conclusion, there has been dramatic advancements in both the understanding and management of PFIC-related diseases, but there are clear challenges that remain, including really lack of effective treatments. Even though the IBAT inhibitors have changed the way in which we approach these patients, we still need more effective therapies. The phenotypic variability, genotype-phenotype modifiers, there's so many ways in which we still need to explore and understand these patients. And then continued idiopathic disease. Again, about 10 to 12% of these patients still don't have a genetic diagnosis despite all of our fancy testing. And so we need to continue to look for other genes that may be available. And as noted, I really think future efforts that combine technological advancements in basic science may enable improved therapies, faster pipelines, and more personalized approaches to the way that we care for these patients. So I think there's about 8 minutes left. I would be happy to take any questions that have come through or any questions that people have. Please pop them into the chat. If not, I want to thank you for joining me. And I think this has been recorded, so you can always go back and take a look. And please reach out to me anytime with any additional questions if there are any. Okay. Not seeing anything pop up in the chat, so everyone gets 5 minutes back in their day. Again, I appreciate everybody for joining today, and hope you have a good weekend. Take care, everybody.